In this last video of the cytoplasmic membrane system of the cell, we're going to be discussing the endocytosis and the transport of proteins or the targeting of proteins from the ER and the Golgi apparatus to specific organelles like the mitochondrion and the chloroplast. So first, we have already learned about exocytosis, how calcium ions are important in order for exocytosis to happen. So now let's talk about endocytosis. So this is when cells internalize cell surface receptors that are bound to extracellular ligands. Okay, so we can see here in this illustration, cross-section of the cell membrane, and we can see how this portion of the cell membrane has sort of started to cave in because there are ligands or the molecules that are detected by receptors that are lining this membrane here, and these molecules are yolk proteins. So yolk is actually food for the cell. And we can also see the coating here, and this is made of clathrin. Okay, and we can see here the formation of a coated pit. So we can call it a coated pit. And we can see here how the vesicle is about to close. And here we have a complete closed vesicle that contains the yolk proteins that are endocytosed, and the lining is composed of clathrin molecules. So this is going into the cell. So this is retrograde transport. And then we also have phagocytosis. So if we differentiate the two, phagocytosis is the uptake of particulate matter. Okay, so not just extracellular ligands, but in this case, bigger particles of matter. So like for example, this macrophage is phagocytosing a yeast cell. So this is phagocytosis. So we need to look at receptor-mediated endocytosis. So again, as mentioned a while ago, there is the formation of a coated pit as shown here. Okay, so this is the extracellular side actually. And you can see here the ligands that have been detected by receptors. So they are all sticking around this pit here. So in this case, these are low-density lipoproteins that contain, that is also known as the bad cholesterol. And this is a cytosolic side. You can see here, so this is the opposite side from the one shown on the left. You can see here a lattice of coating materials, particularly clathrin, coating this coated pit. So this one is the coated pit, okay, with the LDL attached to the receptors. So this is how it would look like. So this is the drawing. So you can see here the coated pit and the clathrin proteins, this scallion-shaped coating that are surrounding the vesicle. So how is this formed? So we can see here, this is the extracellular side, the one at the bottom. So you can see there could have been ligands that have stuck here already, causing the formation of this coated pit. Okay, and the coated pit would be almost completely coated with a clathrin lattice here on the inside portion of the pit. And a protein called dynamin is going to be cutting the connection between the outer membrane and this vesicle, thereby forming this endocytic vesicle that contains the materials for endocytosis. So there's an experiment wherein GTP hydrolysis has been prevented. And without the help of GTP, this diamine ring is just going to continue on forming, and it's gonna cause the elongation of this membrane that we see here. So GTP is needed, GTP hydrolysis particularly, in order for the endocytic vesicles to separate from the plasma membrane. So here's an illustration of the scenario shown here at the bottom. So we can see here in endocytosis, that's from the outside of the cell, we can see the particular ligands. Okay, so there are those purple ligands attached to what we call as housekeeping receptors, the red ones. So these are actually the ligands that need to be constantly removed from the outside of the cell. Like for example, your LDL, the bad cholesterol, needs to be removed from the outside constantly. So you have your housekeeping receptors for that. And then you have other receptors here shown in green for other ligands, perhaps hormones. Okay, these could be hormones. So once the receptors have attached to their own ligands, clathrin will then coat here on the inside, and then the formation of a vesicle here. Okay. And then clathrin is going to detach from the vesicle. And here we can see the vesicle with the ligands and their own receptors. So you have your signal receptor and the housekeeping receptor. Now, the signal receptors are going to be sorted in this sorting compartment, and they're going to be separated from the rest. Okay, because they've done their job. Their only job is to take note of certain signals from the outside of the cell. So they're going to be sorted, and they're going to be destined to be destroyed in the lysosome. So they will be destroyed. While the housekeeping receptor, the one attached or the one that will detect LDL, is going to be recycled back and brought back onto the surface of the cell. So again, the reason for that is the signal receptor has done its job. Okay, its only job is to look for signals, watch out for signals, while the housekeeping receptor's job is a routine job, sort of how a housekeeper does his or her job. It's a routine job. It happens every day. Okay? So that's why the housekeeping receptor is recycled. It's not destroyed. It's going to be sent back to the membrane again. So here's the destiny of your signal receptor. It's going to be brought into the lysosome. And the lysosome is going to be filled with acid, and the enzymes are going to work and destroy the other molecules together with the signal receptors. So this particular process is known as receptor downregulation, meaning to say, if you don't destroy your receptor, your cell is going to be overwhelmed with the signals from the outside, right? Okay? So in order for that to be prevented, the receptors have to be destroyed after they've done their job. So the housekeeping receptors are going to be brought back up again here so that they can continue bringing in the LDL, which is bad for, of course, the total health of our body. So LDL is going to be constantly endocytosed with the help of the housekeeper receptors. So here's an example of a housekeeper receptor, the one for the LDL. Okay, so LDL is known as the bad cholesterol. Presence of it in your blood is highly correlated to arteriosclerosis and, of course, heart attacks and strokes. As we see here, the formation of plaque in the blood vessels is due to the presence of LDL. So LDL is going to oxidize, and the oxidation of LDL is going to destroy or injure your endothelial cell, the cell lining the blood vessel from the inside. And because of that, there's sort of clots that form to heal the wound, supposedly. But the formation of this clot is just going to contribute to the constriction of the opening of the blood vessel, causing blockage in that particular blood vessel, and which may lead to heart attack or stroke. So here's sort of a summary of the bringing in of materials from the outside of the cell. It can be phagocytosis, when the material will be large chunks, okay, as we see here, or endocytosis, or receptor-mediated endocytosis, like the one for the housekeeping receptors and the cholesterol, the LDL. Okay, and here we even have macropinocytosis. So this is like the cell drinking small portions of the extracellular fluid. And of course, the role of the lysosome is central here. So the question now is, how do vesicles know where to dock? So we already explored on how they move, okay, using the cytoskeletal system of the cell and with the motor proteins. We already saw how they are docked onto certain target membranes, okay? But how do they know where to dock? That's the question, okay? So the answer lies with a type of phospholipid known as phosphatidylinositol, okay? With the help of a kinase, the phosphatidylinositol is going to be converted into phosphatidylinositol phosphates. And these phosphatidylinositol phosphates are the ones that sort of line the membranes and are used as signals as to where a particular vesicle is docking. So as you can see in this illustration on the right, different membranes on the cell, both cytoplasmic membranes and endomembranes, have different coatings of phosphatidylinositol phosphates. So here they are represented in different colors. Okay, so a vesicle would know that, oh, this is actually this portion of the membrane because it can detect the type of phosphatidylinositol phosphate present there. And then, of course, the post-translational uptake of proteins. So how are the proteins carried to the other membranous organelles of the cell? So first, in the peroxisome. So the peroxisome actually would be able to get the proteins that it needs from the ER and the Golgi apparatus, because those proteins will have a peroxisomal targeting signal. Okay? And then the peroxisome will also have translocons or channels on its membranes that will allow the protein which contains the PTS, the signal, to enter the peroxisome. And this translocon is so big, is so large, that the proteins can enter the translocon in their native state. Meaning to say, if they're already folded, there's no problem. They can just pass through the peroxisomal translocon with ease. So they don't need to be unfolded again. But the story in mitochondria and chloroplasts is a bit different. So in the mitochondrion, there are 4 possible destinations of the proteins from the Golgi apparatus. What are those 4 destinations? It can be the outer membrane of the mitochondrion. It can be the intermembrane space. That's number 2. Number 3, it can be the inner membrane of the mitochondrion. That's number 3. And number 4, it can be the matrix of the mitochondrion. Okay, so the proteins can either have any of those 4 as destinations. So in the mitochondrion, the protein which is already folded needs to be unfolded again before it can pass through, okay, this translocon of the outer mitochondrial membrane. Okay, so here in number 1, we have a protein that's destined for the matrix. Here in letter A, we have a protein that's destined for the inner membrane. Okay, so they're going to enter the TOM or the translocon of the outer mitochondrial membrane. So they have to be unfolded first. So here on the first scenario, the protein enters through the inner translocon of the mitochondria, okay, to be brought into the matrix because it's destined to go into the matrix. But here on the right, we have the protein that's destined for the inner membrane. So here's another translocon, but this one is going to open laterally so that the protein will just move with the other phospholipids and proteins in the inner membrane of the mitochondrion. Okay, so this is powered by electrical potential, the movements through biased diffusion. And here in the chloroplast has 6 possible destinations for the proteins. And what are those? Number 1, the outer membrane. Number 2, the intermembrane space. Number 3, the inner membrane. Number 4 will be the stroma. Number 5 will be the thylakoid membrane, and number 6 will be the thylakoid lumen. Okay, so those are the 6 possible destinations. So in the chloroplast, we have the outer translocon. Okay, so here for this particular protein, and here on the right, we have another scenario, same process, the protein entering the outer translocon of the chloroplast. Just like in the mitochondria, the proteins have to be unfolded first. And then they pass through the inner chloroplast translocon, which we've already passed through here in the slide. And then this protein on the left is destined to be in the stroma. So once it reaches the stroma, it is folded into its final native shape. But this protein on the right is destined to be in the lumen of the thylakoid. So it needs to enter first the thylakoid, again here, and it's in the lumen where its final shape is shaped using chaperone proteins.